this is working. So I've got my thumb here, or my phone here, just to check that I am connected, and hopefully I am. That's my phone. Okay, so in today's class, what we're going to be looking at is Section B experiments. So, sorry, bear with me. So Section B experiments is what we're going to be focusing on today. And you know that we're going to be looking at question 9, and question 9, we're going to look at the list of experiments that will appear on question 9. And one of these experiments that we're covering today, you will be asked in June 2025, or they might do a mixture of them all. So brilliant, I can be seen. Thank you. Great. Right, so just to go over an overview of the experiments that we're going to look at today. So we investigate the effect of pH on enzyme activity, temperature on enzyme activity, heat duration on enzyme activity. We're going to look at how we prepare one mobilization and examine it, then prepare and show the production of alcohol by yeast, and then conduct an activity to demonstrate osmosis. So they're the ones that we're going to focus on today. Now, I had a piece of paper and I'm not forgetting it. I'm actually just going to step to my desk here for a second because there was something I wanted to mention first. I've left it on my desk. So that's what happens when you go live. Okay, so I'm back. Right. Now, I just want to talk about the importance of enzymes. I'm just trying to find, do I have a space here that I just want to, yeah, this will do for now. Okay, so I've just gone to the last page, and you might have a refill pad there beside you. So what is important is the chemical nature of enzymes is proteins, okay? And that could be a question. At the beginning of an experiment, an enzyme experiment questions, what's the chemical nature of enzymes? So they are proteins. Now, what's the protein look like? So we've covered a protein when we did food and biomolecules, and proteins are made up of carbon, hydrogen, oxygen, and nitrogen. And some of you, sorry, just fix this. Sorry, just another thing. Can I bring this over? There we go, that's a bit better. Okay, so what are proteins made up? What are proteins made up from? So proteins are made up from amino acids. So I'm just going to draw here random shapes. So here's my chain of amino acids. These are making up a protein. Now, so I'm going to rub out this. So with amino acids, hopefully like this make a bit of sense. I have a line here. Okay. This is what an amino acid looks like, NH3. Now, don't be worrying, you won't be asked this. Some of you may be chemists here, but hopefully this will make sense in a moment. Okay, H off that. So if I was to look at this shape here, this is what they would look like, okay? And the bonding here is hydrogen bonding. Here is an enzyme. Okay, and I know that this important part here is the active site of an enzyme, which the substrate binds to. And the active site that slightly changes or alters its shape to accommodate the substrate, to form the enzyme substrate complex to make a product. So this is our active site. Now, anything can affect the hydrogen bonding. Okay, so anything can affect the hydrogen bonding. And what will happen then is your enzyme will start to, so imagine if this is the enzyme, your enzyme will start to unravel. When it unravels, it loses its shape and can no longer carry out its function. Oh, sugar, hold on. Oh, all that has disappeared. Okay, so I'm just going to write the unraveling part. It can unravel, lose its shape, and no longer carry out its function. What's that called? That's when the enzyme has become denatured. I would like students now to say, when they're describing a denatured enzyme, you have somewhere in there the word permanent. So I went from the shapes like this to with our peptide bonds to here's our hydrogen, or here's our enzyme. We describe the enzyme because they're proteins as folded. When they start to unravel, so here's our is folded. When they start to unravel, they become denatured. Okay, so now let's go back to experiment 1. So the first experiment to look at is to investigate the effect of pH on enzyme activity. So what are we looking for here? We're basically trying to find out which pH is the optimum for the enzyme. And the enzyme that we're going to look at is catalase. So here's their stuff. So what's the first thing? Make a little note here. What is your enzyme? Your enzyme is catalase. What's your source of your enzyme? Where did catalase come from? So catalase is the source. So I'm going to write here the source. It's celery. For your enzyme experiments, what is your substrate? Your substrate is hydrogen peroxide. Or you can say H2O2. What's your products? Your products are going to be water and oxygen. So that's your H2O and your oxygen. These two is visible as foam. So your product is water and oxygen visible as foam. Do not say foam. You're going to say water and oxygen is visible as foam. So your enzyme is catalase, your source is your celery, your substrate is your hydrogen peroxide, or you can say H2O2. Your products are water and oxygen visible as foam. That could be the start of introducing an enzyme experiment question. And by the way, you can ask, oh, I'm going to get to the washing up liquid, Sarah, now, literally just now, but thank you. So we're going to go through these experiments. So add catalase, pH 9 and washing up liquid. Back to Sarah's question, what's the purpose of the washing up liquid? The purpose of the washing up liquid is to trap the oxygen that's being released, so we're able to see the foam that's there. Now, you don't need, in biology, I don't know about the other sciences, but in biology, you do not need to say how many grams or centimeters cubed of catalase you used. So you need to mention you will add catalase, pH 9, and washing up liquid into a graduated cylinder. So that's what you're going to do first. And the washing up a liquid, and if I write down here actually, or up here to the side, and that could be a question. What's the purpose of the washing up liquid? It's going to trap the oxygen that's being released and it will help form the foam. So if I write here, you can see that, yeah, washing up liquid. So the purpose of your washing up liquid is to trap the oxygen that is being released which helps, oh, I don't know what's happening there, which helps form the foam, or which forms the foam. Okay, so that's important that you're mentioning catalase, wrong pen, catalase, pH 9, and washing up liquid first into a graduated cylinder. You're then going to put hydrogen peroxide into a boiling tube. And you could say here H2O2. Okay, so you could mention here that this is H2O2. And remember I mentioned earlier just there, hydrogen peroxide, that's your substrate. So you're going to place the boiling tube with your hydrogen peroxide and the graduated cylinder that contains your catalase, your pH 9 and your washing up liquid into a water bath at 29 degrees. Now, the water bath at 29 degrees is the optimum temperature of the enzyme catalase. Optimum, what does it mean? So optimum means that it's the most efficient, which I'll get to now in a moment. Now, why am I putting the boiling tube in the water bath? Because your boiling tube of hydrogen peroxide would have been handed to you maybe in a syringe. That's what I would do in school. I'd have that syringe, and that has come from the fridge or the freezer. That's where your hydrogen peroxide has come. Fridge. I'm thinking of getting confused with ethanol. Fridge. So we want to bring, if it's come from the fridge is around 4 or 5 degrees Celsius, we want to bring the hydrogen peroxide up to the same temperature. So you're going to place your boiling tube and your graduated cylinder in a water bath at 25 degrees. How do you maintain the temperature of your water bath? Okay, so you can set it to 25 degrees. So you're going to wait for both to reach 25. And then when both have reached, so you put in a thermometer, and when both have reached 25, you're going to add your boiling tube of hydrogen peroxide into the graduated cylinder. Now, when you're mentioning your boiling tube with your hydrogen peroxide, slowly, and I've put it in bold, you're adding the hydrogen peroxide down the side of your graduated cylinder. Okay, you're not putting your hydrogen peroxide into your graduated cylinder. 0. You're slowly adding your hydrogen peroxide down the side of your graduated cylinder. That's 3 marks. Okay, so that difference there, that could be a point, that's 3 marks there. Sorry, I'm looking here because I have the phone, so if you have any questions, please just ask them there. Now, the moment you add your hydrogen peroxide, you're immediately going to record your volume, and this is called your initial volume. So you've taken them out, you've taken out your graduated cylinder, you've taken out your test tube of hydrogen peroxide because they've reached 25 degrees. You're going to slowly add your hydrogen peroxide down the side of your graduated cylinder, and you're immediately going to record the volume. Then you leave that there for 2 minutes. So what technically is taking place is your enzyme catalase, with your substrate hydrogen peroxide, is going to join up and become the enzyme substrate complex. And what the active site of that enzyme catalase is slightly changing shape to accommodate the substrate, to break that substrate down and form a product. So after 2 minutes, what you're going to slowly add the hydrogen peroxide down and calculate the height of the water and foam. So this is called, please write in here, initial volume. I have to look, I'll on the next one. So you call the initial volume. After 2 minutes, you're going to record the final volume. And the final volume is the height of your foam. But we never mention foam. Okay? We might say it in class in foam, but in an exam, we're not going to mention foam when we're talking about a product. It's oxygen and water visible as foam. So to find your rate of enzyme reaction, it's your initial volume from your final volume after 2 minutes. And those 2 minutes is actually a point in that. That's 6 marks if they ask you, how did you measure enzyme activity? So I'm going to actually put this here, calculate rate. I can't spell today. Calculate rate or enzyme activity. So it's going to be your final volume minus your initial volume after 2 minutes. If they ask you this question is 6 marks. The first 3 marks will be going for final volume minus initial volume. The second 3 marks will be going for the time that you referenced. Okay, and it has to be a set time. Don't say after time, 2 minutes. So if you're doing this experiment, you're not going to leave it there for 5 minutes. Realistically, we don't have time in a lab to do that. So it's for 2 minutes. So then what you would do is because the experiment says investigate the effect of pH. So that is what our independent variable is. This is what we're changing. So we're going to then repeat the exact same. So you can say repeat using pH 4, 7, 10, different pHs. I would use 4, 7, and 10. So I did start with 9, which is our optimum. So a few things here with this. And again, I'm going to write on the side. What is our control? Your control is here at the bottom. Your control is boiled catalase. What you have done is you have denatured the catalase. So what your teacher might have done is got a sample, and what I would do is I just have a small bit at the start and I'd say, look guys, I'm looking after the control. This is what the control is. I've denatured the enzyme. So your control is your boiled catalase, or you could say denatured enzyme, denatured catalase. And if it's denatured, it has lost its shape and can no longer carry out its function. Now, somewhere in that sentence, write the word permanent or permanently. So you could say at the beginning, it's permanently lost its shape and can no longer carry out its function. A denatured enzyme has lost its shape and can no longer carry out its function. It's permanent. Somewhere there, please put the word permanent. Because once the active site of the enzyme is denatured, that enzyme cannot revert and go back to what it was before. Okay, now a few things with this. So we've said our enzyme is catalase, the source of the catalase is celery, our substrate is hydrogen peroxide, our products are water and oxygen visible as foam. Our water bath. How do we maintain the temperature of the water bath? So temperature here is a factor. There is factors in enzyme experiments that are kept constant. So I'm going to push this over here. So factors kept constant. So we need to know these factors we keep constant and how we keep these constants that they may ask us. So factors kept constant. So one is a water bath. So how do we keep, sorry, that's not water bath, that's temperature. How do we keep temperature constant as we use a water bath at a set temperature? And you can say 25 degrees, because in this one, optimum temperature for it is 25 degrees. So set temperature, set water bath, or you could say a thermostatically controlled water bath. Set water bath at 25 degrees Celsius. Okay, what was another factor that we kept constant? And another factor that we kept constant is substrate. Our substrate was hydrogen peroxide. So how do we keep hydrogen peroxide constant, the same throughout this? And that's when I use pH 4 and 7 and 10. And that's because you use the same volume of your, so substrate concentration. So we use the same volume of substrate with each little experiment we do here. So I'm going to say substrate concentration. That's our second factor. When we use this, how do we keep it constant? So that's the same volume of substrate. The third factor that we keep constant is enzyme concentration. How do we keep the enzyme concentration constant? How do you keep the catalase volume constant? We use the same. So same volume of enzyme. So there's three factors in this experiment here, investigating the effect of pH on enzyme activity catalase that we keep constant. We keep the same. Temperature, how? Water bath set at 25 degrees. Substrate concentration, how do we keep it constant? Same volume of substrate. Enzyme concentration, how do we keep it constant? Same volume of enzyme. Okay, a question they may say is, how would you, and I'm going to write this down, how would you vary the pH? So vary the pH by using different pH buffers. So maybe I'll try it this way. How do you vary the pH? Okay, so use different pH buffers. So you could say the question, how did you vary the pH? I use pH 4, 7, 9 or 10. Or you could say use different pH buffers. And then, or you could say use different pH buffers, example. Okay, so I want to make sure that I've covered. So that's all within our setup. Now, in 2024, they were very strict regarding giving the marks out for experiments, and they wanted different things linked together. So I'm going to go back over this and the important things that are required to investigate the effect of pH on the enzyme activity. You must mention your catalase, you must mention pH, and you must mention your washing up liquid. That those three first are added into your graduated cylinder. Your experiment is not going to happen. Oh, wrong colour pen, wrong colour highlighter. Your experiment is not going to happen if they're not there. The second thing is you must mention what your substrate is, your hydrogen peroxide. Then you must mention together, please preferably, that your boiling tube of hydrogen peroxide and your graduated cylinder are placed in a water bath at 25 degrees. So, for example, if somebody said place a boiling tube and graduated cylinder water bath, or place a boiling tube and graduated cylinder into a water bath, they may not get the mark because you haven't said the temperature. They're getting very strict now. They don't like giving away H1s. They might want, so if someone said a boiling tube was placed in a water bath, but because they didn't have the word graduated cylinder, they may not get the mark. So link these together is what I'm trying to say. So you're mentioning boiling tube, graduated cylinder, and water bath at 25. You will have noted here previously I've mentioned that you've had your hydrogen peroxide in the boiling tube. So this implies step 3, that the boiling tube you're referencing here is the boiling tube that has hydrogen peroxide. So you want both to reach 25 degrees. Take the boiling tube out and the graduated cylinder out. So they are now out of the water bath and they're on your workbench where you're conducting this experiment. Very, very, very important. I've mentioned this. Slowly add the hydrogen peroxide down the side of the graduated cylinder. So something similar was asked last year regarding DNA isolation, and the students had to add ethanol. And what we were looking for is slowly add the ethanol down the side. Now, if students said slowly add the ethanol, they got 0. So slowly add the hydrogen peroxide down the side of the graduated cylinder. See, missing one word, 0. Okay, so slowly add the hydrogen peroxide down the side of the graduated cylinder. If you said add the hydrogen peroxide down the side of the cylinder, no, because you didn't say the word slowly. Okay, so just that would be one thing that's sticking out of my head from 2024 is how strict they were with students on the experiments and the detail that they wanted. Okay, so we've added that, and now we're just about to start our experiment by you're going to calculate out the height of the oxygen and foam, or calculate out, you can say here, the initial volume. Okay, leave it then for 2 minutes. There has to be a reference to time. Okay, there has to be a reference to time. And then after 2 minutes, calculate out then your rate of reaction. Calculate out the volume of foam. So your final volume minus your initial volume after 2 minutes. And then repeated this using different pHs. So my approach to this would be, if asked about pH, is read the question carefully. See, are they asking for the method? And if they're asking for the method, what are they asking for underneath? Because underneath, and if you've been looking at past paper questions on enzyme experiment, they may ask you, how did you record the rate of reaction? What did the hydrogen peroxide act as? What was your product? What was a factor kept constant? How did you keep this factor constant? So read that before you go rushing in and answering straight away. What other information do they want from you as well? Now, the other information that they would want from you, there would be no marks if you repeated that on your method. Okay, and then also you're using a very valuable space in your answer, because section A is handed up, section A and B is handed up in a booklet, and that's what's scanned. Well, that's one of the other things that's going to be scanned. What happened there? Okay, if you're asked to draw a diagram, please make sure that you reference what the temperature is. Now, I know I have here steps, but what I actually don't have here is I don't mention that this is, I changed my pen, this is my hydrogen peroxide. In my graduated cylinder here, I don't say, and I'm going to do it now, and if you add in 2, in here is my catalase, in here is my pH 9, and my washing up liquid. So you can actually put a line through this steps that's there. Okay, so the next page is showing your possible results. They're not going to ask you what were your results. They're not going to ask you for results like this where you have figures. So it might find what was the optimum enzyme for this activity. And it might be so somebody might have used pH 9 and somebody might use pH 10, or somebody might use pH 10 and not 9. So you should find that pH 9 or 10 was the optimum enzyme for the catalase. Why? As it produced the most product. They may ask what is meant by the term optimum. Okay, so that is a possible question near the end of the experiment. And optimum means that the enzyme is working at its most efficient. So the enzyme excuse me. Now, this little thing here, I dropped my pen. This little thing here is just showing you, here's our enzyme catalase. Our substrate is going to join up with your enzyme catalase to become your enzyme substrate complex. This is where the active site of the enzyme has slightly altered its shape to accommodate the substrate and is forming a product at as fast as possible rate. And at the end, our active site here is going to go back to its original shape. The enzyme stays the same. And now we have our product. What is your product? Oxygen and water visible as foam. Okay, so oxygen and water visible as foam. That's your product. They may also ask you to draw a graph of your results. So remember, when we're working with pH, we have a narrower range. Okay, so if I was to draw this freehand here, this would be my result. If you're asked to draw a graph, there'll be 3 marks for labeling the pH on the x-axis and 3 marks for labeling on the y-axis. So I have enzyme activity here, or you could say rate of enzyme activity. Actually, add that in, please. Rate of enzyme activity. And our enzyme activity is measured in centimeters cubed. Or you could say enzyme activity here and put centimeters cubed. Actually, I'm going to say use the word rate, because anything to do with rate is to do with time. And what we looked at here is that we added the hydrogen peroxide, we left it there for 2 minutes. So do include the word rate. So remember, in enzymes, controlling all factors is very important. We mentioned about controlling the temperature by using a water bath set at 25 degrees. We mentioned enzyme concentration. So that's, we use the same volume of our catalase, and substrate concentration, we use the same volume. And be very aware, all enzyme experiments are controlled, like temperature has an effect. So be very, very aware with all enzyme experiments, temperature has an effect. If it's too hot, it'll denature the enzyme where it's lost the shape and can no longer carry out its function. This is a permanent. So, and it's the active, it cannot be reverted back. Do I need to have a title on the graph? Will I lose marks if I don't? Very good question, Sarah. No, you don't. On these graphs here, you don't need a title. But what is important is you label your X and your Y axis. For labeling your X and your Y axis, that alone is 3 marks. And then for drawing your graph, That part is 3 marks. I would not ignore this experiment if I was sitting my Leaving Cert in 2025. I would not ignore this experiment. Okay, so this might actually appear on your mock. If I was setting my mock for my school, which I'm not this year, but if I was, I'd be making sure that somehow that's appearing on the mock paper. If not in section B, in section C. Because just remember as well, folks, you could be asked an experiment question in section C of your paper as part of a long question, or in those lovely questions now in 16 and 17 that are worth 30 marks. I can get a 30-mark question of this experiment and put it into section C as well. Do not ignore this experiment. Now, I've done quite a lot of detail there, but the detail there that I've done and I've spoken about is going to carry us through when we look at our next experiment, which is to investigate the effect of temperature on the rate of enzyme activity. So, do you need to write the number of pH on your graph? No, Aaron, you don't. Okay, good question. If I go back here, no, you don't. And if you look, and I don't have them here, I have the enzyme, sorry, what am I saying is I don't have enzyme past paper questions in this, but if you look at the enzymes chapters in section B, at those questions, you literally just a narrow range. Because if you had to do, back to your question, Aaron, if you had to put in the letters, sorry, in the numbers of your pH, you would have looked at pH, so let's start off. I know pH 7 is in the center, so this would be pH, so you would have looked at pH 4, I'm really running out of space, 10, so your graphs would be like this. No, not what pointy. Does that make sense? So you just need one narrow range. Okay, now, so what I'm going to do is I'm going to rub this off, because if this is recorded and people see it, they might think, so this would be your graph here. Now, let's investigate the effect of temperature on the rate of enzyme activity. So in this one, we want to find out what the optimum temperature is for the enzyme catalase. And in this one, we already know what the optimum pH is, and that's pH 9. And that's because we've done the previous experiment. So the same things that I would write here. What's your enzyme? It's catalase. What's your source of your enzyme? It's celery. What's your substrate? It's hydrogen peroxide. What's your product? Oxygen and water visible as foam. And then it's the same thing. So what we want to see, or what we want to find, is which temperature produces the most product, the most foam. So let's talk through this one. And again, I'm going to say as well, guys, that let's be clever about this. Let's include the key points in our method if they ask about the method of this. So again, if you're asked about temperature, read what they're asking you first. See what's underneath the main method or your procedure part, because if you're repeating yourself and it's you're not going to get the mark in your method part if it's part of the procedure. If they ask about at the end, what's your substrate? So add your catalase, your pH 9, and your washing up liquid to a graduated cylinder. And the same purpose of what's the purpose of your washing up liquid? It's going to trap the oxygen that's being released so it's visible as foam. Our substrate is hydrogen peroxide. The same thing, we're adding hydrogen peroxide to a boiling tube. We're going to stand our graduated cylinder and in a boiling tube in a water bath at 0 degrees. So I would do 0 degrees first. That's my ice bath. So I would have got a basin of either ice and a bit of water. So around 0 or 4 degrees. Again, when they both, so then at 0 degrees, are you going to get your hydrogen peroxide up? No. So you slowly add the hydrogen peroxide or ice cold hydrogen peroxide. Don't need to be ice cold. I'm going to bracket that off. Slowly add the hydrogen peroxide down the side of the graduated cylinder. Then the minute you add your hydrogen peroxide really is when your experiment is starting to happen, because it's your substrate. So you have put your substrate in contact with the active site of the enzyme catalase. So what you're going to do is then you're going to record your initial volume. Okay, so you can say calculate the height and the height of oxygen and foam, or you can say calculate the initial volume. record the initial volume and then the final volume. And you can say, leave that there for 2 minutes. And then you can say, record the final volume. Oh, that sentence doesn't make sense. I'm going to add in here, record the final volume. Do you know when you sit and you make something and you read it and read it and read it and read it and go, yeah, I'm really happy with that. I was really happy with that. And now, as I'm seeing with a fresh pair of eyes, a period later, I'm like, it doesn't read well. Does that make sense? Is I use it? Yes. Is it okay if I use liver instead of celery? Yes, it is okay if you use liver. That used to be part of the old experiment, but you can, Sarah. Yeah, yeah. Because liver is still catalase. So yes, Sarah, you can. So record the final volume after 2 minutes. Okay, so then you're going to repeat this at different temperatures. So I have here 25, 40 and 60. So some of you may have had in and around 20, 25. You might have gone 40, 60, you might have even gone higher. So the same thing, how would we vary the temperature? So that could be a question. So I write here, question, vary the temperature. So we'd vary the temperature by using different water baths at different temperatures. So I'm just checking there. So here is this. And you could say I varied the temperature by using different water baths at 25, 55, 65. Do I know as an examiner what temperature you used? No. Are you giving me the answer I want? Yes. What do I do? Thank you. 3 marks. Next question. Okay. What are our controls for this experiment? The same as before. Your controlled is your denatured enzyme, your boiled catalase. So this is your control, your denatured. And I'm writing here again, denatured. What does denatured mean? It means that the enzyme has permanently lost its shape and can no longer carry out its function. Now, let's look at factors that will be kept constant. Will I fit it this side? Yeah. Factors kept constant. So we're going to have 3. The first is pH. How do we keep the pH constant? We used pH buffer. But we used pH buffer 9. So used pH buffer 9. Do you have to say 9? No, you can say pH buffer. And some of you may actually have used pH 10 as your constant pH there. And again, that's to do with what have you got available, what have your teachers got available in their labs. The second thing that was kept constant is your enzyme concentration. How do we keep this constant? Same volume of catalase. And our substrate concentration. I'm just going to CONC for short. And how did we keep that constant? The same volume of hydrogen peroxide. So there are factors that are kept constant, and these are the 3 factors that are kept constant here. What did we change in this one? What was our independent variable? It was our water baths at different temperatures. And you're trying to figure out which water bath, which temperature produced the most product here. So here is our control. We're mentioning here, if you're asked again to draw a labeled diagram of your apparatus that you used, do it like this, please, please, please. Don't do, so as I would say to my students, I'm laughing here now, I would say to my students, sometimes this is what I get. I get a test tube, I get a graduated cylinder, I might get here randomly a water bath, I might get here ice cubes. I'm like, I don't know. I don't want to know what the ingredients are. I want to know what does the cake look like at the end. Does that make sense? So you're not, what is the setup? I don't want to know what goes into it. I want to know what is the final setup. So if you're asked to label your diagram that you drew, let's say if this is my graduated cylinder, what is in it? So I have my pH 9, I have my catalase, and I have my washing up liquid. And I forgot to label that this is your graduated cylinder. Here is my water bath, and my water bath is set to 0 degrees. Here I have my hydrogen peroxide in my test tube here. Oh, I'm going, and yeah, it's one thing that annoys me in correcting experiments is when I get the ingredients and I don't get the cake. Okay, that's the easiest description of me saying this. I don't want to know the bits. I don't want to know. I just want to know, what does it look like? Okay, and your teachers in school want to know, what does it look like? Okay, so we've mentioned our enzyme, our substrate, our product, what the purpose of our washing up liquid, and what our control is. So again, we found out what is the optimum. So optimum is the most efficient. Okay, so we mentioned optimum before, and I showed this before. Now, here's your graph of a temperature. So you need to be able to recognize this graph or draw this graph. You need, again, the same thing is your x-axis temperature, and you could say degrees Celsius, and I'm going to write here, rate of, I'm liking the word rate, of enzyme activity. I could put in my unit. So these together, 3 marks. Now, with a temperature, your range is much wider in comparison to the pH. So the pH graph was something like this. So my range was narrow. I've got a problem with that word, narrower. So with your temperature, it's wider. Hopefully that makes sense. So if they asked you to draw that literally, that's what you're drawing when you're labeling that if asked about this. And reminding you again, in enzymes, controlling all factors is very important. Okay, we want so in this one, what we controlled was pH, our enzyme concentration, and our substrate concentration. What we were changing was our temperature, because we wanted to find out what's the optimum temperature for the enzyme catalase. Oh my God, I just can't believe the time. Oh, right, okay. I'm really focusing on enzymes here, guys. My gut is quite good, and my gut tells me enzymes is the one in question 9 this year, or a mixture. Okay, now denatured enzyme. highlight, ding, 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 ding. Denatured enzyme is important. Okay, so you're investigating the effect of denatured heat denaturation on the enzyme activity. So in this one, your control is your unboiled catalase. Okay, so your control is your unboiled catalase. So let's look at the steps. Again, we're adding our catalase into a boiling tube at 100 degrees, and we're showing how we're denaturing it. What is denatured? So if you're asking about this, and you're going to ask you, what does denatured mean? The enzyme has permanently lost its shape and can no longer carry out its function. Add the boiled catalase, because our experiment is investigate the effect of heat denaturation. Add your boiled catalase, your pH 9, and washing up liquid to your graduated cylinder. You're going to stand your graduated cylinder and your boiling tube hydrogen peroxide in a water bath at 25 degrees. When your hydrogen peroxide comes up to 25 degrees, because it's come from the fridge, you're then going to slowly add the hydrogen peroxide down the side, and I'm going to get the bracket off again, ice cold. Slowly add the hydrogen peroxide down the side of the graduated cylinder. And we're going to calculate out our initial volume. So calculate out the height and foam. So you're going to record the initial volume and then the final volume after 2 minutes. Make a reference there that you're leaving it there for 2 minutes. You're going to repeat this procedure with your unboiled catalase, which is your control. Your diagram. And make sure, if asked about this, that you clearly show of your graduated cylinder here. I have here, this is my unboiled catalase. This is my boiled catalase. My unboiled catalase, this is my control. In here I have my pH 9, my catalase, and my washing up liquid. pH 9, because we've already found out it's the optimum. pH 9, because we've already found out, is the optimum pH. 25 degrees, because we've already found out it's the optimum temperature for this enzyme activity. And this what's added in here looks like this is your hydrogen peroxide. And I'm just going to label this graduate cinder. Okay, what factors are kept constant here? So I'm going to write down factors kept constant. I'm not going to put in the how because I've said that twice already and I'm sure you've gotten it down already, but I'm just aware of the time. I got carried away. Okay, factors kept constant. Okay. First off, we have temperature. And temperature is kept constant as we're using a water bath set to 25 degrees. pH is kept constant as we're using pH buffer 9. Substrate concentration, we're using the same volume of hydrogen peroxide. And the fourth, enzyme concentration, we're using the same volume of the enzyme catalase. And then this diagram is here. So your boiled catalase did not produce any oxygen or water. Your unboiled catalase, it had all the ideal conditions. It had a correct pH, the correct temperature. So it did produce oxygen and water visible as foam. Now with this, if they do ask about heat denaturation, I'd put my money on it that they're going to ask you what is the control in this experiment, and your control is your unboiled catalase. It might ask you, how did you denature the enzyme? And you make sure you reference, you boiled it at 100 degrees Celsius. If you just said, I put it in a water bath and let it boil, we're not going to give you the marks, okay? Because you might have originally boiled that to 100 and the temperature might have gone down. So make sure you say at temperature if it asks of how did you boil it. So I would really have that, mainly pH and heat denaturation on my radar for my mocks and for your summer 2025. I haven't seen any mock papers yet, not released, but I think this, and sorry, I'm just checking for a question. So do not ignore this. If I was sending my mock paper this year, I would put in either heat denaturation or pH in section B on question 9. And the other one, whichever one I did not use, I would put that as in question 16 or 17 as a 30-mark question. That's what I would do. And that's what I would do if I was setting a mock, but I'm not. But that's what I would do here. In my school, what we do is we would make our own mock papers based on what we think will appear in the summer. So if any of you Black Rock guys are listening, that's what we're going to do this summer. That's still going to happen. Sorry, not for the summer, for your mock. Right, immobilization. Now, immobilization was asked last year, so I'm going to go through this quickly. While I'm going through this quickly, it does not mean that they cannot ask you in section C what is meant by the term immobilization, given examples of immobilization, what's the advantages of immobilization. So I'm going through this quickly, but they can still ask you those are the other terms about enzymes in the experiment. Sorry, about enzymes, immobilized enzymes in section C of the paper. So this year with this, we wanted to see students mention sodium alginate and water and yeast together, mixed up together in a beaker. And a reference somehow that your yeast contains the enzyme sucrase. So they were all mixed sodium, blah, blah, blah. Now, here's another mistake I made. In a beaker. Oh, I don't know what's happened there. So in the summer, students got a mark for mentioning the piece of apparatus a piece of apparatus somewhere in here, but we wanted to see sodium alginate, water, and yeast first to get the mark, to get the 3 marks that were there. So you're going to draw this mixture into your syringe. You're going to release from a syringe 1 drop at a time into a beaker of calcium chloride. And what the calcium chloride, what you're going to is you're going to leave those beads to allow to harden in that calcium chloride solution. So what actually immobilized the enzyme is the sodium alginate with the help of the calcium chloride. Now that was asked a couple of years ago, 2016, 17, and students didn't do well on that. Then we're still not finished this part. We're then going to filter the beads through a sieve. And then I get my students then just to rinse it through the tap with water. And that's because if I have my immobilized enzyme here, I might have some leftover yeast stuck on the outside. And if we're putting through the water, what we're doing is you're getting rid of that excess yeast, because that could have an effect on your experiment results. So this is broken up into 2, the preparation of immobilized enzymes and the examination of immobilized enzymes. So that's what we're going to look at now. So with your examined immobilized enzymes, we have our immobilized enzymes in beads. Your control in this is free yeast. So this is no sodium alginate. So it's your free yeast mixed with water. And in here I have 2 set up here my retort stand, not labeled. You have your dropping funnels. Again, not labeled, please label. And this is your claw of your, claw tap. And I've labeled glucose test strips. So into your 1 separating funnel, you're going to put your immobilized enzymes. I said dropping funnel, I meant separating funnel. I have to fix that. Your immobilized enzymes. Into your control, you're going to put that free yeast solution. To both, you are going to add sucrose. Sucrose, is the substrate here. So we're going to add sucrose, and the product that we want to get is glucose. So you're going to test for glucose. You can say with glucose test strips or with clinic strips. And you're going to repeat this at short intervals or 2-minute intervals until your glucose appears. So for us in school, it's the little purple mark. It'll start going brown, bluey first, and then a little purple strip. Okay, so then you're asked to compare the turbidity of the solutions. So that's the cloudiness of the solutions. And what you would find is, so note that the glucose test strip was turned quicker in our control, in our free yeast. And that was because the enzyme was not immobilized. Now, what I have added here is your main conclusion here is immobilized enzymes, they are going to produce a pure product of glucose. The advantage of using immobilized enzymes, and I've actually done this in class before, is at the end of the experiment, I will reuse those immobilized enzymes again, just to prove that this actually works. Or else I'll use some of those mobilized enzymes for my next 5th year class. So last year, 2 5th year classes, and I actually went in between the both. This is showing that this is an advantage of immobilized enzymes, is that they are reusable. Now, it took longer in the immobilized to test for glucose, but your immobilized enzymes are easy to recover, to take them out, reusable, and you're a pure product. So what you're getting here, this glucose is a pure product here of glucose. And another advantage of immobilized enzymes is that they are more stable than a free enzyme. And again, temperature is an important factor in these experiments, as enzymes are sensitive to temperature changes. And this is linking back with pH, the effect of pH, the effect of temperature, and the effect of heat denaturation. Because what can the temperature do? Can denature. Okay, so we're finished with enzymes. Please, please, please go over enzymes. Right, now, I just have the notes here. We are on to light intensity, to effective influence of light intensity on the rate of photosynthesis. So what you need for this is you need a plant, and the plant we use is Elodea, but you could say Canadian pondweed. So you're going to place your LOD into a boiling tube with pond water. So whatever water your teacher would have got the pondweed from, that water will go inside the test tube. And your teacher may also cut the ends, and I should kind of show here, no, not great. I would cut the ends of the stem and have a little pinch, okay? And that's to try and help oxygen bubbles to be released. So you can say Canadian pondweed instead of LOD. Will they accept? I think they'll even bracket off now Canadian pondweed. I think they'll accept pondweed. But if I'm going for my H1, I'm letting them know exactly what it is. So place LOD in a boiling tube with the pond water. Okay. Now, what your teacher may do as well is, or what you may do, is add some sodium bicarbonate, and that maintains a constant. Constant CO2 is needed there. You're going to place this. So you're going to place your test tube or boiling tube with your LOD and the pond water in a water bath, and your water bath or a beaker. Okay, probably I would use a very large beaker, and in my beaker, the water here is just normal water from the tap. So you're going to place then this into the water bath at 20 degrees, and we're going to switch on our light source. And when we switch on our light source, we're going to give about 5 minutes for your pondweed to get used to it. So you're going to place the boiling tube at different distances measured at 20 centimeters. So here's my ruler. So I put it at 20 centimeters with the light source here, leave it there for 5 minutes. After 5 minutes, I'm going to start counting the number of bubbles that are being released. So some of you may have started this at 100 meters and then work your way towards, or start towards the light source, or some of you, excuse me, may start at 20. So the main thing is you're going to leave the plant there, or leave your boiling tube there for at least 5 minutes to adjust. And then you're going to start counting. So then you're going to record the number of bubbles per minute. Repeat this twice. And this is important. This is our rate of photosynthesis. We're recording the number of bubbles and repeat this twice. Then we're going to calculate out the average number of bubbles released per minute and repeat at different distances. You could say, how could you vary the distance? And you could say set distance, meter stick with set distances at these distances here. Now, sorry, pain in my face, pain in my eye. Your graph. If they ask you to draw the graph, or they might ask you to label the graph. Some of you may have looked at this and done CO2. I would use light intensity, so light source. So if some of you do mention CO2, if you are looking at the effect of CO2, you're putting CO2 here. So your x-axis and your y-axis must be labelled to get your 3 marks. That's your light intensity and your rate of photosynthesis. If you're asked to draw your graph, this is what we're looking for here. Now, if you're asked to explain your graph, this is the answer we want. As the light intensity increases, the rate of photosynthesis increases. So I can see here, as the light intensity increases, so does the rate of photosynthesis. That is 3 marks. The second point here is what is happening here. So what's happening here in the blue is the rate levels off as the saturation point is reached, which means that we cannot produce any more food. So this could be 12 marks, as in 3 marks for labeling my x and my y axis, 3 marks for my graph, for my conclusion or explaining my graph, 3 marks for this first sentence. As light intensity increases, the rate of photosynthesis increases. Or I mentioned there, some of you did CO2. As the CO2 increases, the rate of photosynthesis increases. This then, as the rate levels off as the saturation point is reached. I don't know if it was, I think it was pre-COVID or maybe just after we first examined. We wanted this graph explained, and not many at all gave this second point there. And okay, so that's your photosynthesis. I can't remember when this clip. Is it okay if sorry, yeah, same question. Okay, now let's look. Show and prepare the production of alcohol by yeast. So this was in 2023. So I'm not going to, the chances of putting them this 2023 before some other enzyme experiments that have not been asked for a while is a bit slim. So to show and prepare the production of alcohol by yeast, again, this is broken up into two parts. Procedure first. What are we going to do? Our preparation. The first thing that you're going to do, or your teacher might have done, is boiled the water and allowed it to cool first. And what the boiling of water does, it removes the oxygen that's present. And then to that cooled water, or the cooled, boiled water, your teacher would add your glucose. You would then add some yeast to that glucose, and that could be conical flask A. And to conical flask B, you're going to add cool boiled glucose to flask B, but no yeast. And this is your control. You may put, and I would, put a layer of oil on top of each, and that's to prevent any oxygen getting in. Then you have your fermentation lock, and into your fermentation lock, you add lime water. And what lime water will do is test for the presence of CO2. You would place these in a water bath at around, I think it's like 25 degrees. Actually, I forget the temperature. I'm blank. Is it 25 degrees? Yeah. 25 to 30 degrees, and you leave it in the water bath, and you'd leave it overnight, and you'd want that to bubble. So what I would do is, whenever I do this experiment in school, I'd hang back a bit until I see it bubbling, and I will. And what I do is I'll take a little recording and I'll show it to my class and prove to them, look, this is it is bubbling. If it's bubbling, fermentation is taking place. We're making alcohol. If it stops bubbling, fermentation has ceased. We've stopped making alcohol. So to show the presence of alcohol. So the next day, following that day, coming back in. So your teacher might do some of this prep. And what you would do is you take the contents of your conical flask and you would filter it. So we want to get some filtrate. So you're going to filter the contents of each into a test tube. And then you're going to test for the presence of alcohol ethanol. And this is your iodoform test. So you don't have to say potassium iodide and sodium hypochlorite. You could say iodoform. That's your test for alcohol. The second part of a test for alcohol is you then have to place these into a water bath at 60 degrees for 5 minutes. So you filtered it. Here's my filtrate. I've added my potassium iodide and my sodium hypochlorite, which is my iodoform test. Okay, so we can say our iodoform. And then we're going to put that in a water bath at 60 degrees for 5 minutes. And then what we would hope to see is, if alcohol is produced, we'd hope to see clear to pale yellow crystals have formed. And I'm putting here again, temperature is an important factor in this experiment because enzymes are sensitive to temperature change. Okay, and the last one is to, demonstrate osmosis. Now, this experiment hasn't been asked as a full long question in a long, long time, but they may ask parts of this question in question 9. They could ask parts of this question in section C. So what did we use? So remember osmosis. If you're asked about osmosis, you're mentioning the word water 3 times. It's the movement of water molecules from an area of high water concentration to an area of low water concentration across a selectively permeable membrane. So in this experiment, you would have got 2 strips of viscine tubing, and your viscine tubing is acting as your selectively permeable membrane. And you would have placed those 2 viscine tubing in water to soften them so you could open them. Into one, you're going to put a sucrose solution, and into your control is distilled water, or you could say water. And then you would eliminate some air bubbles. So now you have 1 viscine tubing with sucrose and 1 viscine tubing with distilled water. What you would then do is make sure you pat them dry, and then you're going to record the mass of both, and you're going to observe the turgidity. So I washed off any excess sucrose on the outside and dried them. I recorded the turgidity of it, and I recorded the mass. And then you're going to place both into a beaker of distilled water for at least 15 to 20 minutes. 10 minutes, you're not really going to get a reaction, or 5 minutes. At least 15 to 20 minutes, you're going to leave them in the distilled water. After that, what you're going to do is you're going to take out your 2 Viscon tubings and you're going to pat them both dry. And then you're going to record their mass and observe their turgidity. And what you should see is the Viscon tubing with the sucrose increased in mass and, I can't emphasize this, turgidity. Your Viscon tubing with your distilled water, your control, remained the same. Now, this experiment, it could appear, or it could appear in section C, mixed up with diffusion and osmosis, been asked. So guys, that is our masterclass on question 9, emphasis on heat denaturation and pH. My gut is telling me that they may make an appearance in section 9 more than other experiments, or an appearance on your Leaving Cert paper. But that does not mean ignore the others. If you're going for a H1, you know it all. Okay, so you know all your stuff. So that's question 9. Next week is going to be question 10. And next week, like this week, I'll make sure the notes are uploaded by the end of the week, and we'll go through question 10. So hopefully then, for some of you, whether it's your Christmas exams or your mock straight after Christmas, you're prepared to get that 15% on section B. So listen, if there's any more questions, please ask. But have a nice evening. It's getting dark out there now. And tomorrow we're live again, just to remind you, 5:15 to 6:15, we're covering viruses tomorrow. So I'll hope to see you then. So thank you very much.